The topic for the present video is D50 and span in particle size distribution of drug substances. In this video, we are going to learn for D50 and span, what is D50 and span, how exactly these two terminologies are related to PSD, and how to interpret the data. Then we will see why D50 and span is important in PSD. So, whenever you are having the data of PSD, always see the three tier data, that is D50, D90 and D10 data. The PSD, that means particle size distribution, is a very important critical material attribute of the drug substances because it affects the physical and chemical properties. It affects the safety and efficacy of the formulations. D50 and span are two important parameters. Those help to describe the distribution of the particle size within the given sample. Always understand that the powder is made up of different number of particles with different sizes. So to understand the data of PSD, we need to have understanding for D50 and span. D50 is median diameter. D50 means the median particle size in the given particle size distribution. It means 50% of the particles population are smaller than the D50 value and 50% of the particles are bigger in size than the D50 value. It is the particle size at which 50% of the particles are smaller and 50% are larger. In other words, it is the particle size corresponding to the 50% of cumulative distribution. For your easy understanding, consider D50 as 50% of the population or particle population is having smaller diameter or smaller particle size, and 50% of the population of particles is having higher particle size than the D50 value. Interpretation of the D50. So if you plot the cumulative distribution curve of particle sizes, D50 is the value on the x-axis where the curve crosses the 50% of the mark. For example, if D50 is 5 micron, it means 50% of the particles in the sample are smaller than 5 micron and 50% of the particles are having particle size larger than 5 micron. Coming to the relevance of the D50 value in drug development, D50 or median diameter particle size is important because it is the median diameter. It is the central measure of the particle size which can influence the dissolution rate, bioavailability, and overall performance of the drug in the body. So D50 value is related to the safety and efficacy of the pharmaceutical formulation. A smaller D50 value often suggests fine particles, which may lead to faster dissolution and absorption. And in turn, this faster dissolution and absorption affect the Cmax, AUC, and Tmax, and that's why affects the safety and efficacy. So bioavailability and bioequivalence is also related to D50 value. Coming to span, whenever you are having the data of particle size distribution, always understand about span. Span is a measure of the width of the particle size distribution, or you can say it quantifies the spread of the particle sizes within the sample. Whether the powder is consisting of particles with varying particle sizes, whether the width of the particle size distribution is broad or narrow. If the particle size width or PSD width is narrow, that means the powder is containing the particle sizes which are similar to each other, or there is less variation in the particle size of the particles in the powder. Span is calculated by the present formula D90 minus D10 divided by the D50. So in this formula, D90 is the particle size at which 90% of the sample's particles are smaller. D10 is the particle size at which 10% of the sample's particles are smaller. And D50 is the median particle size, as we have seen in the present video. How to interpret span? So span provides you the information regarding the width of the particle size distribution. A lower span value indicates a narrower particle size distribution, meaning the particles are more in uniform size. So narrow or lower span means the particle size is uniform, and if span value is higher, then it indicates broader distribution, meaning there is a greater variety in the particle sizes. Relevance in drug development. The span provides insight into the uniformity of the particle size, which can affect the consistency of drug release, stability, and manufacturing processes. A narrower particle size distribution or lower span is often preferred for certain formulations. Lower span value leads to more predictable behavior during dissolution and absorption. So this is the information regarding span. The example of span I have included here. Consider D10 2 micron, D50 5 micron, D90 10 micron. So D90 minus D10 divided by D50. 10 minus 2 divided by 5 will give you span value of 1.6. So 1.6 is a moderate spread. If the values of span are lower, then means the particle size distribution is uniform. And if it is more than the moderate spread value, it means the powder contains the particles which vary in the particle size distribution. That means the range is higher. Why D50 and span are important? So you might have come across the in vitro bioequivalence. Some of the formulations are required to meet the D50 and span, because if you meet the D50 and span of the different formulations and compare these with the reference formulation, then there are high chances that the formulation will behave similarly in the in vivo. D50 and span both are very important for PSD distribution. D50 helps to assess the typical particle size and is a central point in understanding the drug's behavior. Span offers insight into uniformity or variability of the particle sizes, which can affect the performance characteristics such as drug dissolution and bioavailability. Both of these parameters are critical whenever you are optimizing the formulations, maybe IR release, maybe controlled release, whenever you are working on to the solubility enhancement, and thus ensuring consistent quality in the final drug product. So D50 and span values are required to be considered for any PSD data whenever you are working on to the formulation development. This was the small video regarding D50 and span in PSD for drug substances. Many videos are available onto the Pharma Learning in-depth channel. So I request you to go through those videos and you will have good understanding for these topics. Thank you for watching the video.